Good morning, everyone. My name is Nidhi Vashishta, and I'm a product specialist scientist at ACD. Before we begin the webinar, I have a couple of announcements. Most important of which is that the audience will be muted for the course of this webinar. If you have any questions, please use the chat feature on your WebEx program, and my colleagues from the support team will be ready to stand by your answers in real time. In case we have missed a question from you, we will follow that up offline. This webinar will be recorded and will also made available on ACD website shortly, so please stay tuned. With that, I would like to begin the webinar for BaseScope Duplex Assay. In this webinar, we will be covering the following topics with the technology introduction and overview, introduction to the duplex assay, data review of the duplex assay, and a few applications where this assay can be used. To begin with, BaseScope assay is a unique product from ACD, which is based on the same proven and established RNAscope technology. It's a powerful assay that utilizes the ZZ probe design, which can vary from 1 to 6 ZZ. It provides single molecular detection and is very effectively used for detection of exon junctions and short target sequences. The BaseScope assay is currently already available as a single-plex assay on manual and automated platforms. Today, we are launching the BaseScope Duplex Assay as a manual assay. With this, I'd like to get to the overview of the duplex assay workflow. The assay workflow of BaseScope is similar to our existing platforms, which is RNAscope and BaseScope single-plex, in which you essentially permeabilize the tissue or the cells which are bound on slides. After that, you hybridize the target-specific RNA. Here, you can detect 2 targets with the BaseScope Duplex Assay. So you hybridize the target probe 1 and target probe 2 here. On top of which, the signal is amplified using the modified branch chain signal technology from ACD. And the signal can be further seen using a bright field microscopy as green and red chromogen deposits. With this, I'd like to get into the probe design overview for the BaseScope assay. As you can see that the BaseScope probes are designed for customers' gene of interest or any target of interest that you may have. We use the proprietary probe design algorithm to design and identify a specific region where we can design the probes, which takes into a lot of other considerations of the unique region, the GC-rich region, and we'll cover approximately between 50 to 300 nucleotides for the BaseScope probes. The standard probe configuration varies from 1 to 6 ZZ of oligo pairs covering 50 to 300 nucleotides. If it's a unique exon junction detection, the probe design will mostly be a 1ZZ probe detecting the unique junction. And if it's a short target mRNA, the probe design will vary from 50 to 300 base pair. With this, I'd like to get into the details of BaseScope probe design. As you can see on your screen, the BaseScope probe can be depicted as oligonucleotides in shape of Z, where the lower region of the 2 Zs together forms the target-specific binding site, covering a 50 base pair region. Each Z only covers 18 to 25, but together they cover the total 50 base pair region. There is a spacer arm connecting the top and the lower region of the Zs. The top part of the Z is a unique region, which actually forms the preamplifier binding site. And as I mentioned, the BaseScope probes can vary between 1 to 6 ZZ depending upon the target length and the sequence of interest. Today, we have over 1,600 peer-reviewed publications using our unique and proprietary RNAscope and BaseScope assays. This shows the increased market adoption of our technology and its unique utilization. So far, we have the following portfolio of the assays that we offer: RNAscope and BaseScopes, which can, depending upon the target of interest, you can have a chromogenic single-plex or a chromogenic duplex assay, or if you're interested, you can have a multiplex fluorescent assays. If you have short target of interest, up to between 50 to 300 base pairs, you can either utilize the BaseScope single-plex assay or the BaseScope duplex assay. For the duration of this webinar, I will be focusing on the BaseScope duplex assay only. The BaseScope duplex assay is recommended for co-expression or mutually exclusive expression in two different cell types. As you can see by the schematic on your screens, you can detect two different targets in the same cell, or you can detect two different targets in two different cell types. However, this assay is not recommended to detect two different regions on the same transcript. And this is not recommended because there can be a lot of competition. With this, I'd like to get into the chemistry of the BaseScope duplex assay. Here, this schematic is depicting the chemistries. As you can see, there are two independent amplification trees that are built for each channel. For the C2 tree, we utilize the alkaline phosphatase and the fast red chemistry, whereas for the C1 tree, we utilize the green chromogen and the HRP chemistry. These two trees are built on two different targets of interest. And this is essentially a workflow for the BaseScope duplex assay. I would like to focus you on the top headers, which are pre-treatment, hybridize and amplify, and counterstain and mount. The assay is divided into three short parts, which includes pre-treatment, hybridization, amplification, and counterstaining. Pre-treatment would vary depending upon the sample type that you are using. So please connect with our technical support if you have any questions regarding sample pre-treatment, or you can download the user manuals from our website. The assay in total takes about 11 hours to run and has two different color detection, the red color and the green color detection for each target of interest. So once you've done this, the data should look something like this. As you can see, specific green and red dots for control probes, which are housekeeping genes. Here it is shown on cell pellets. Something like similar you would also see when you're using tissues. As you can see, individual green and red dots indicating that the housekeeping mRNA can be detected. These are some applications of the BaseScope assay. As you can see, that BaseScope assay can be used for detection of the exon junctions, the splice variant isoforms, gene knockout, circular RNA detection. All of these are very short targets, and usually we will design a 1ZZ probe for detection of such unique exon junctions. And for short targets, you can detect highly homologous gene sequences. You can detect small nuclear RNAs. You can detect gene editing through CRISPR and pre-microRNA sequences as well. With this, I would like to show some specific data regarding the BaseScope assay, as this assay can be used for cell type specific knockout validation. Here we have a specific knockout where we are trying to detect in wild type versus knockout mouse. So in the wild type mouse, we have a cell type of interest, and in that particular cell type of interest, we have knocked out a gene of interest. So as you can see that the green here represents the cell type marker, and the red in here detects the gene of interest. So what happens when you're taking the wild type mouse tissue and trying to detect two of these targets together, here for marking the cell type, we have the SST probe and the P-Val-B probe, and the gene of interest is named as gene A. This assay was done through our service team, and you can see some clear cell marker detection using the SST and P-Val probe. Here it is marked by the green arrows, and the gene of interest, which we are calling gene A, can be seen with the clear red dots. However, what happens in the knockout mouse, we are able to detect the green signal, which targets the specific cell of interest, and you can see clear green detection. However, there is no gene of interest detection as that has been knocked out. So this is a very good assay to validate your knockouts in cell-specific regions. Another very good application of Basescope Duplex is CRISPR-mediated gene editing. As you know, CRISPR-mediated gene editing can affect the gene at DNA level, or it can affect the expression in multiple ways, such as insertion, deletion, or point mutations. You can utilize almost all the assays from our portfolio in detecting such gene editing. RNAscope assays can confirm the target or downstream gene expression. The BaseScope single-plex assay can actually detect the one single edit. However, the BaseScope Duplex assay can be utilized in a very novel way where you can identify the gene edits in a specific cell population by duplexing a cell marker with the edit specific probe, or you can detect both the wild type and the edited probes at the same time. That's something what we have done. As you can see here, this was our design for using BaseScope Duplex assay for CRISPR-mediated deletion. The deletion was of 49 bases only, and you can see that the 49 bases on the left side of the figure has been depicted with the green bars, and the region right adjacent to those 49 base pair is depicted as red bars. So what happens on the right hand side, you can look at the probe design. So for detecting the wild type transcript, the probe was designed exclusively in the 49 base pair region, whereas for the edited transcript, the probe is designed right outside the 49 base pair, and it detects the unique junction that is created after the editing. So as you can see, we first qualified the samples by using the housekeeping genes, PPIB and polar 2A. And as you can see here, you can see the green dots depicting the PPIB mRNA, and you can see the red dots depicting the polar 2A mRNA. The right-hand side panel is the negative control, which does not show any signal. So that shows the assay background is very well controlled. So here you can see the unedited and the edited, two different sample types. This is done specifically in mouse liver. And the gene that we are trying to edit, we're calling it gene A. And this is we're just looking at the liver samples. And as you can see here, in those liver samples, the hepatocytes show on the right-hand side quite a lot of edited mRNA. And on the wild type, or on the left-hand side, the wild-type liver shows only the wild-type mRNA. So you can see here that cells have, on the right-hand side, have biallelic as well as monoallelic edits. The wild-type hepatocytes are highlighted by the black triangles on both the images. However, only on the right-hand side images, you see the white triangles that show both the biallelic, edited mRNA. In this right-hand side image, you also see hepatocytes that are actually co-expressing both the alleles, as that has been shown by the black arrows. Interestingly, our assay also provides unique morphological information that this gene editing was not detected in around the cells that were around portal vein, the bile ducts, and the central vein. So this basically provides a morphological landscape of the tissue to show where the CRISPR gene editing is actually happening. The Basescope Duplex Assay, as I said, can also be used for unique splice junction detection. And here I have one such example by detection of MET. MET receptor actually goes through a lot of mutations. There has been a lot of reported mutations of MET receptor, which most of them involve the deletion of exon 14. So what it creates is the MET14 delta transcript. And using our BaseScope probe design, we have tried to detect the unique exon junction, the exon 13, exon 15 junction that was created only in the deleted transcript, and MET14, 15 junction that is present in the wild type transcript. And we've done all of this assay in cell lines. You can see here, we have two different cell lines. The top panel shows the wild type on the left-hand side, and it shows the MET14 delta cell lines on the right-hand side. And as you can see, each of these cell lines are exclusive for the probes. Wild type is only showing the 14-15, the wild type probe, and the MET14 is showing the exon skipping probe. And in the lower panels, we have actually mixed these two cell lines, and we've still been able to detect the wild type and the exon skip and the alternate transcript. And similarly, on the right-hand side panel, lower right-hand side panel, you can see both the transcripts. And in that panel, you can actually see more of the wild type transcript because that has been mixed in a 5-to-1 ratio. Also, to show that this effect is not based on the chromogens or the assay chemistry, we repeated this experiment by switching the probe type. So now the wild type is detected in red color, and the MET14 delta is detected in the green color. And as you can see, similar to the previous result, each of the cell lines are exclusive for detecting the wild type or the MET14 deletion transcript. And in the lower panel, you can see the 1-to-1 ratio mixed cells also show clear signal, and the 1-to-5 ratio mixed cells show more of the wild type transcript. We also ran a background control check on these cell lines just to show that whatever we are detecting is specific signal and is not the background of the assay. As you can see that the DAB-B control was completely clean for these cell lines. Once again, these are some applications of the BaseScope Duplex Assay. You can combine this with any existing BaseScope probe that you have. You can also combine. You can also detect the circular RNA versus linear RNA in your cells of interest. You can detect circular RNA with the cell marker of interest. This can be the BaseScope Duplex assay can be used for detecting splice variant A and splice variant B in a specific cell. You can also detect short targets from same gene family, and you can detect the edited and wild type gene as we've done in our CRISPR experiments. Here is some information about getting started with the BaseScope Duplex assay. The Basescope Duplex Reagent Kit contains pre-treatment reagent, detection kit, wash buffer, and target probes. Control and target probes are C1 and C2 probes. Usually the C2 probes are provided in 50x concentrates. This is some ordering information for Basescope Duplex reagents. As you can see, we have intro packs available for human and mouse tissues. The intro packs actually include the control probes and control slides as well. We have some duplex control probe packs, which are species specific as well. The recommended accessories for the Basescope assays are shown here. The hybridization system and easy batch slides can be purchased through your ACD account managers. This is a checklist for the Basescope Duplex assay that shows the reagents and the materials that are provided from ACD. Also, the checklist contains the reagents and materials you will need from your side. You can get all of this information on ACD website. To get the information for the Basescope Duplex product, you can go to our products page and you can find all the related information about this assay and other products on the product page. To get the user manual, you can go to the user manual page on our support website, and you can use our interactive drop-down menu selection for finding the